To begin our story, we need to go back in time. A time where life on young Earth began to evolve into more complex organisms. 2 billion years ago, towards the start of Proterozoic era, endosymbiosis, one of the biggest breakthroughs in the history of life, gave birth to the mitochondria. Just imagine, one day in the distant past, one bacterium started living within another larger bacterium, and what began as a chance encounter between two microorganisms ended up making all complex life on Earth. The theory mitochondria originated from bacteria may not be familiar to everyone. One of the evidence supporting this theory is mitochondria has its own DNA, which is circular and closely resembles bacterial DNA. As we all know, nuclear genetic stability is very important for the progression of life. In the event of a genomic instability due to DNA damage, the nucleus is designed to respond by either repairing the damaged DNA, arresting cell cycle progression, and even killing the defective cell. Like the nucleus, mitochondrial DNA is also susceptible to damage. Mitochondrial DNA damage is associated with a lot of diseases, such as neurodegenerative and age-related disorders. As much as we know how important healthy DNA is for a functional mitochondria, and how essential a functional mitochondria is to keep us alive, there is very little information about how mitochondria respond to DNA damage. Dr. Colin Campbell's lab is devoted to excavate the answers to this question. His lab's primary focus is to understand the DNA damage response, especially what kind of repair mechanisms the mitochondria prefers.
So DNA damage repair pathways are less understood in the mitochondria as opposed into the nucleus, and one reason for this is the unavailability of experimental assays that can be used to validate the mitochondrial response after DNA damage. Our lab has developed a novel experimental assay that can be used to validate the DNA damage response in mitochondria. We use electroporation to introduce damaged DNA into mitochondria. Previously, people have used electroporation to introduce DNA into bacterial cells and into mammalian cells, but no one has electroporated damaged DNA into mitochondria to study the DNA damage response. Basically, we use this machine to apply an electrical field to isolated mitochondria. Since the mitochondrial membrane can't pass current, it acts as an electrical capacitor. Subjecting membranes to a high voltage electrical field results in their temporary breakdown, resulting in pores that are large enough to allow DNA to enter. We developed this assay that can be used to study double-strand break repair in the mitochondria. In the nucleus, it's known that double-strand breaks are repaired either by the homologous recombination pathway or by the non-homologous end joining pathway, but it's unclear which of these pathways is responsible for double-strand break repair in the mitochondria. To answer this question, we built this plasmid, and in it we have an insert for the 475 base pair insert that's homologous to the mitochondrial genome. In this insert, there is a recognition site for a restriction enzyme called APA1. In the backbone, there is a recognition site for an enzyme called PAC1. So then we can either cut it with APA1 or with PAC1. By cutting it with either of these enzymes, we'd be forming a double-strand break and we'd be forming a linearized plasmid. We then take these plasmids, electroporate them into the mitochondria. This APA1 site is in the region that's homologous to the mitochondrial genome. This was only repaired in the presence of a homologous donor. This site here does not have an available homologous donor, so we can then allow it to incubate for a few hours, recover the DNA, and then use it to transform bacteria. Only healthy circular DNA will transform bacteria, so only if this break or this break has been repaired will we be seeing colonies formed after the transformation. So after doing this assay, we'll transform the bacteria, count the colonies, and look at the transformation efficiency and compare it between these two plasmids, and that will give us insight as to what mechanism is responsible for the repair of double-strand breaks in the mitochondria.
So what? Who cares? Why should we invest our resources in understanding DNA damage responses in the mitochondria? What new insight would that give us about the mitochondria? To answer that, let's go meet the chief himself.
So the research that Maram is pursuing is focused on a narrow question, which is trying to understand specifically how DNA double-strand breaks are repaired in mitochondria. It's part of a larger project that I think has a greater and deeper impact potentially, and that is, what is the relationship between the accumulation of DNA damage or the failure to adequately surveil and repair DNA damage and human health? There's an intriguing correlation between the accumulation of DNA damage in mitochondria in our cells as we age. So we know that as people get older, their tissues accumulate higher levels of DNA damage in mitochondria, and we see the sorts of changes that are associated with aging. The hair turning white, loss of muscle mass, failure to adequately function with respect to memory and learning. And the question is, are these DNA damages the consequence of the aging process, or are they driving it? So I would say one of the fundamental questions in biology that remains unanswered is, what causes aging? If I showed a group of people pictures of 10 people, there would be a consensus. Who's the young one? Who's middle-aged? Who's old? And yet we don't understand precisely what drives that process. Now, my lab's not interested in trying to cure aging, but we'd like to understand the drivers of it, and we think looking at the dynamics in mitochondrial DNA and the loss of integrity of mitochondrial DNA may be playing an important role. We'd like to be able to learn more about that process in an effort to potentially improve quality of life in people as they age.